Good morning, children. Yesterday we have started chapter number 6, molecular basis of inheritance. Two types of nucleic acids are DNA and RNA. What are functions of RNA? What we have to discuss in this chapter is replication, transcription, and translation. Then, what is the number of base pairs present in different individuals? What is the Watson and Crick model of DNA? We have done. Now the question was why the distance between two polynucleotide chains of DNA always remains constant, because always a purine pairs up with a pyrimidine. Now today's topic is what do we mean by central dogma? The concept of central dogma was given by Francis Crick, which states that the genetic information flows from DNA to RNA to protein synthesis. This is called central dogma. First of all, we will write the concept was given by Francis Crick. What does it mean? That genetic information flows from DNA to RNA to proteins, and then we will draw this diagram. That is, this shows formation of DNA from DNA. This process is called replication. Then DNA, it transcribes messenger RNA, and the process is called transcription. According to the sequence of nitrogenous bases in on mRNA, proteins are synthesized, and this process is called translation. Now, next question is, in some viruses flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to the process? We have seen yesterday that in some viruses RNA is the genetic material. So what happens in those viruses? RNA first undergoes uh, reverse transcription process. देखिए, otherwise formation of messenger RNA from DNA, this process is called transcription. Now if the reverse takes place, that is formation of DNA takes place from RNA, this process is called reverse transcription, and this is possible because of presence of an enzyme, that is reverse transcriptase. So the question is, in some viruses flow of information is in reverse direction, that is from RNA to DNA. Can you suggest the name of the process? So what is the name of the process? It is reverse transcription. Now, packaging of DNA helix. We have done that distance between two base pairs, consecutive base pairs in DNA is 0.34 nanometer. That is equal to 0.34 into 10 raised to power minus 9 meter. If we want to calculate the length of DNA, how the length of DNA is calculated? It is equal to total number of base pairs multiplied by the distance between two base pairs. Now say if we want to know the total length of DNA, that is haploid set of human DNA, then we know the total number of base pairs is 3.3 into 10 raised to power 9. That is the total number of base pairs. So what is the distance between two base pairs? It is equal to 0.34 into 10 raised to power minus 9 nanometer. So if we multiply this, what will be the total number? It will be around 1.1 meter. But if we want to calculate that length of DNA, in a diploid cell, so we will multiply by it by 2, so it will become 6.6, and it will be approximately 2.2 meter. So length of DNA is 2.2 meter.